up. This is actually how we work together in R&D to make data science and AI a reality. So Thomas has been, he's a physician by training, has been in the pharma industry for many years, decades even, pharmaceutical companies, smaller, larger, also worked in contract research organizations, so he knows actually how it feels to have customers, which I think is really interesting. And maybe the most interesting part, before joining us, Thomas was actually heading the Danish regulatory agency, so he knows also what regulators would like to see in this space. So, and now making the jump on data and science and development. Lars has been with Novo Nordisk for 27 years now, I believe, somewhere around this. A protein chemist, so actually many of the things you got excited about today, you know, dare I mention amycretin, come originally from Lars's group. You know, this is where all our good chemists sit and make these genius molecules that we put to work in humans. But then actually Lars felt there is the next big thing, and this is digital science, and this is why we call it digital science and innovation, because we believe it will not only help us to do things better, it will help us do things that we can't even imagine today. Welcome, Thomas.
Thank you so much. And thank you so much also, Markus, and the rest of the executive management team for being good warm-up band for us today, because you set the scene, and I hope you can understand why Lars and I, hand in hand with our teams, would like to take you through why we do this, why we are so focused, and why we invest so heavily, and why we believe that we are very well placed to introduce or to further invest in data science and AI. You've seen the enormous amount of data we have generated in our outcomes trials, in the research, in the clinical part. You see what is coming. You see the pipeline grow. So just because of that, the volume, the explosion of number of trials, number of data points, data modalities, just for that reason, AI is necessary. At least if you want to do it in this century. Human beings cannot anymore analyze clinical data, multi-omics data, image data in due time. It's simply too big. You have heard, at least most of you have heard about AI for sure the last 12 months because of OpenAI. But we have been working in Novo Nordisk for quite some time on this. You can see it on the right here. We already started heavily looking into this in 21, and then we have really intensified our investments. I'm going to take you through why we believe we have the exact right building blocks to deploy AI across the value chain. We'll focus in on R&D today, but this is across the company. And as you can see here, we actually are deploying it already across the value chain. Lars will dive into what he's doing in research. In development, we are deploying it. I'll give you some concrete examples of what we are doing. It's in trial design, etc. But why we're doing this, it's more speed. You heard my boss, Martin Lange, say, you know, we want to reduce the cycle time. So faster, and it can be done faster. Scalable, we can handle more data, more clinical trials. Quality by design, you know, I'm not saying computers are perfect, but they're more predictable in their errors and you can find them. So deploying AI will increase quality and more insights, which you talked about as well. You know, we talked about the select data, I'll dive into it, but that's just one part of the database, of the totality of data we have in Novo Nordisk. So across in Henrik's area, scaling up to the size he talked about today does actually require AI. All the new operators are trained with virtual reality before they're actually in the factory. Just one example. They also deployed in the assembling lines for quality inspections and so forth. And then we have a foundational layer that goes across. To successfully deploy AI, most importantly, I will say actually they're all important, but you need the right capabilities and skills. And Lars and I have been investing heavily in attracting the best in the field. And luckily, it's easier now than it was 5 years ago. And this is not just computer scientists and engineers and coders. We also need to link them up with the biology scientists, with the physicians. Without the right technology and infrastructure, without your data house in order, AI will remain AI with no added value. Many people don't really think about that, but you need to get your data house in order. And then the data. So it's not just having the data house in order, it's which data do you have? And we have, as of today, the largest, deepest cardiometabolic database of any kind. Adding to that real-world data, adding to that the research data, adding to that a lot of other types of data. And you start being able to analyze that, image data, omics data. You can imagine opportunities of repurposing, discovering new targets, new molecules, optimizing your trial designs, and so forth. That is one of the main reasons we want to build this. The other one, as I mentioned, is speed. So this is trial execution from study startup, from automated data ingestion. The analytics, even the statistical code, which is also in my group, will soon be done 50, 60, 70% by AI, and reporting, and I'll come back to that. So this is really why, and we are well placed. Now I'm going to show you some of the very, very exciting areas that we are not just on a PowerPoint slide, it's being built. We have a minimum viable product this year. This is Found Data. Found Data will be and is being developed as the infrastructure, cloud-based infrastructure of the totality of clinical data first, and then we'll add largest data on subsequently. From more than 1,600 randomized controlled trials being put on the platform over the coming months, year, it's not decades we're talking about. We already have many trials on there. Harmonized, standardized, adding images, multi-omics data, and all the data mentioned before. This will make it easy to find real time. It will make it easy to interrogate, and in addition to that, and analyze with low code and no code. So that's amazing. The last thing I want to mention here is a couple of examples before I'm out of time on how we deploy AI right now. One is Novo. This is our automated AI-powered report writing machine, if you may. And this has already been used for submission of study protocols study reports to FDA. I'm taking out a piece of paper here because FDA assessors and EMA assessors have said to us, based on more than 9 reports that we have submitted, removal of subjectivity is welcome, and the NovoScribe version is clearer and more succinct, easy to review. So the regulators are happy. When they are happy, it goes faster. This is being scaled. And just be careful of the details here. We have reduced the writing times from 90 days to less than 10 days. And by next year, we expect a lot of FTE reduction. So there's productivity in this as well. Another example is our AI adjudication. When we do all these large outcomes trials, traditionally all these events have to be manually adjudicated by physicians. We have an algorithm now which will reduce adjudication time from more than 30 days to minutes, and costs from approximately 15 million kroner per trial to 2 to 3 million kroner per trial. And that cost reduction will go up as the model advances. So these are a couple of the examples. By this, I want to hand over to my excellent colleague, Lars, to take you into the early part of the AI space.
Thank you, Thomas. He almost deserves a small applause, I think. So applying AI to predict in a better way who will get hit by chronic disease carries value. I think that is rather obvious. What is maybe not that obvious is how we can use the same AI to tease out new targets and biomarkers. It's actually the same AI that can do this. Together with Microsoft, we have deployed an AI system on what we call the UK Biobank data. Marcus showed you a slide earlier on with all the cohorts that we are actually purchasing these days. Using the UK Biobank, we use the medical records of the population, but also use genetics, proteomics, metabolomics, and so forth. All of these data go into our training of the AI. This is not a large language model. We want a model here that do no hallucinations. That's important. Henceforth, it's really important to collaborate with somebody who got the model and know how to apply the models. We use this to come forward with a better model to predict cardiovascular disease. Now, because we have the proteomics in the model, we can actually look at, or the AI can find the correlations between what proteins goes up and down in this large data set, and can find correlations to new targets. Sometimes we can always drive it as far as drive it into causality and actually say this is the gene who actually are driving this disease. On a really lucky day, we also get a biomarker associated with this, and that, of course, will drive value in terms of increasing the probability of success when we move this into our pipeline. So here is an example of value for the patients, because we can predict who are most likely to get hit by chronic disease. Value because we can generate more new targets and biomarkers. Let's move on to another example that we are doing right now. This is designing molecules, designing peptides. This is more my home turf, you can say. When we design a new molecule, it's important that we scout the landscape of which one will be a better molecule. Once we lock the molecule, the atomic structure, we can do that only once, and then it lasts forever. The atomic structure of semaglutide actually encompasses all the things that you heard of today in the molecular structure. So the more we can actually screen, the better molecules we can find. So what we have done here is to take a peptide called calcitonin as a scaffold and turn it into an amylin selected compound. That has never been done before, driving sort of new innovations. We can always have a discussion on who will be most innovative going forward. Will it be the AI or will it be the humans? In this case, it's a combination. But what we did here, we generated 250 million new molecules in silico and screened those in 4 different models. Henceforth, we are up to 1 billion evaluations. That is many orders of magnitude higher than what we could do with the old methods. And that actually is driving this new innovation where we can generate super high amylin selectivity out of a calcitonin molecule. We also did it without doing a lot of compounds. We made 2,500 compounds, and that's not a lot. On a normal day in a normal program, we'll do 10,000, 20,000 compounds. To educate and really tap into the real world data, are we on track? But actually we're doing less compounds when we do use these methods here. So the key point with this AI is that we're driving innovation, but we're also driving efficiencies, because we only needed 3 design cycles to come up with this fantastic compound, whereas we normally would use 6, 7, 8, 9 design cycles to do this. So innovation and efficiencies are what we're driving using the AI in molecular design. At Novo Nordisk, we have a long track record in trying to use as few animals when we do experiments in research and development. I'm super happy that we can actually throw AI into this mix. I think that carries the value alone. When we do animal experimentations at Novo Nordisk, we use the highest standard, the highest ethical standards to do this. So it actually takes time and resources to do proper animal studies. And that's why when we can eliminate those or go into a bare minimum of experimentations in animals, we actually save a lot of time and resources. In this case here, we were able to model an entire biological system, so we could predict the pharmacology of the compound. Moreover, we were also able to do a model so we could predict the pharmacokinetics, how long time the compound would rest in the body, and actually rank order our compounds in these 2 model systems without using animals. That's time saving and resource saving, and henceforth also good because we don't use that many animal models. So these kind of methods are speeding up our research. In this case here, we also did highly advanced screening. I want to give you the biggest number today. 10 to the 13th molecule were interrogated in this. That's 10 billion, no, trillion. I just wanted to surpass any number that you've heard today with this. And that is what we're capable of doing. What we are doing is finding a really fantastic mini binder, replacing an antibody, henceforth scalability. And this is what we can do. This is my most exciting program where we can do AI for the molecule and AI for the screening and teasing out the right molecule. We cannot do it alone. So we are seeking partnerships to drive AI at Novo Nordisk, all from the foundational technology bits and pieces, but also into the science. I want to highlight Velo because they're driving target discovery based on a really fantastic data set, real world data. And on top of that, they have built a AI flywheel that allows us to find new targets and promote them faster than ever before. We are not done. Thomas and I and the rest of the company, we will also explore, can we go into larger alliances with the tech companies? We will try to interrogate that. We also foresee that the next bottleneck might be access to GPUs. So we need more hardware to actually train our models. Before it was access to talent. Thomas addressed access to high quality data, but we also need to ensure that Novo Nordisk get access to GPUs. Finally, we are getting our toes wet, working with quantum. A lot of the problems that we're dealing with in chemistry and peptide design are actually quantum problems. So in summary, I hope we have taught you a little bit about how we actually in our daily life apply data science and AI to promote our pipeline. We're talking about innovation, efficiency, and quality. And actually sometimes also just our employees having more fun on concentrating on the really fun parts of the job. We have a really rich source of data, especially our clinical data, and we are also utilizing external collaboration to drive this. So with that, I will give the word to you, and I think Marcus, you'll try to control this.
Yeah. Thank you, Lars. Please. Thank you. So open for questions, and we have microphones in the room, so online participants can. Should we start with Michael? I'm not as sophisticated as Carsten to maneuver the Q&A, so one at a time. Yes, please.
So Michael from Nordea, maybe a question on sort of the development cycle and the optimization of time. So writing an application, etc., so it decreases significantly. So if you take from reporting of phase 3 data now to filing, what kind of optimization of time should we in total expect to be able to be done with new technologies?
So there is the sprint, as you are alluding to, where everything, but let's not forget all the upstream where you can save out. So where we are optimizing is all the way from study startup, more innovative study designs by clinical trial simulation, which we do routinely now. You can reduce and optimize the design of the trial and test, can we get the same result with much less visits? Can we do more decentralized, etc.? So that's study setup. Then there's the study execution. The more you can survey and do risk-based approaches, the more you can shorten recruitment time and the more you can shorten execution time. Then you have the whole statistical analysis. I mean, today, we are deploying a system which will automate the entire data flow, not without humans in the loop, but including statistical analysis and reporting. And then you have the submission in itself and the review. May I just take a few seconds on that? What takes time today, both in US and Europe and elsewhere, is the review time. You know, the reviewers today do manual review. They cut and paste. We want to, and we talk to our tech partners, to walk towards a much better submission. So to answer, so that's the long answer. I don't think we're talking about days. I don't think you're talking about weeks in reduction. I don't want to give you an exact, but our ambition is north of months in reduction of cycle time.
Thank you. Please.
Thank you, Florence Espedes from Société Générale. Quick questions. What do you do differently versus your competitors? What gives you an edge? Is it your database from your internal clinical trials? And what could you do better, let's say more to be even better? Could it be investment in IT, in arts, or more collaborations? Maybe one last, it's a bit naive question. When I see 8% improvement on your table, where there is a lot of noise behind artificial intelligence, I was a bit surprised by this number. I would have expected something better, but it's just a stupid naive question.
You want to take the
let's take that. That is actually highly significant. And just to know, it is not something that I think is actually being submitted to a high-ranking journal, the Microsoft team and also the Novo team. So what you should look at here is also that the model will only get better. Right now, the sort of only data we have thrown into the model is proteomics data and the medical record. And so just expect when we throw in metabolomics, other data, we will see higher predictive value coming out of this. And also, we have just only added the UK Biobank data in the example I show you here. When we add the other cohorts, expect these numbers to go up. So that is something where we'll see the models progress over time, just like you've seen the chats going from the GPT chat 2, 3, 4, and hopefully soon 5. It's a little bit the same with these models that we're building here.
I'll answer the first part of your question very briefly. So the main differentiator from us and our competitors is the quality and the size and depth of our clinical data and our preclinical data. There is no other company today. There's no other who has the depth, and you just saw the SELECT study alone, but now start to combine SELECT and 1,600 randomized control trials. So that's one. Then there's our infrastructure. If there is a server left in this organization, those who run it will seek them out. It's entirely cloud-based, and it's completely seamless. So it's designed to be seamless. All the processes can be automated and can be scaled. And I know because we have a Disrupt is a network that I co-founded with J&J and AstraZeneca, a co-chair, which is 12, 13 SVPs of the biggest pharma data science organization. So we have benchmarked ourselves. I know exactly where we are in investment, and we came from a place where we weren't in the top. And in the last 2 or 3 years, we have moved into in many areas where other companies look at us and say, this is the leading. The competitive edge will not just be the models, it will be how you deploy them and the data you have in. So this is some of the answers.
Yeah, just behind you. Right. Yeah.
Thanks, Seamus Fernandez at Guggenheim. So wanted to get, I think, to Florent's question, a little bit of the, learnings from hallucinations, things like that. What are the benefits as you kind of process, this movement from AI-based molecules over to sort of wet lab, processing? I'm assuming that's a part of the education. And then the second question is, on the data science side, I did notice that the CagriSema trial is, the CV trial is designed for only 7,000 patients. How much of the AI dynamic has actually kind of played into that with your own learnings from the SELECT trial as a benefit?
So maybe I take a step on the first one. Actually, we in some cases we would like to avoid the hallucinations, and that's why we use different models to actually increase the accuracy of what we do. In the molecular space, we might not be concerned about hallucinations. We might actually want them to get it one step further out than what we were thinking. And we will cover that by actually tapping into reality, by combining the in silico predictive methods, by going into the wet lab and actually make, say, 1,000 compounds to actually check what of the hallucinations are really useful and which are not, and then retrain the model as such. So I would say in certain cases we would favor hallucinations and test them up against the reality, and in some cases, especially dealing with the clinical data, we would actually do our utmost to apply models where this is not the case.
I can just build on that around, and this is a general, very, very important area that is not very mature in many places. We are not deploying AI across the value chain without doing this in a very ordered and governed manner with trustworthy AI framework being deployed. And I would say we are probably in the industry leader there. We have a complete data and AI governance that we are being rolled out without stopping innovation in the non-GXP area, but being clear that there's no shadow AI in other areas. And in a few years, you have the EU Act coming in, so there are many legislations to follow. With respect to trial designs and trial size, what I mentioned about clinical trial simulations, which we run, we simulate the large trials thousands of times and get full data sets, and then we look for what happens if we tweak here and tweak there. And that's the way to optimize the trial design. In the future, and today it's without images and omics in, and that's the next evolution. So we get really, really exact simulations as early as we can, update the model and run it. And this we do already routinely across the. So these are the reasons why you're going to see hopefully smaller trials. And the question earlier today about what about placebo? So the next step is of course digital twins and virtual trial data. Here we do need the regulators and payers to dance with us, so to say. We can't just march ahead when you're in GXP space.
Yeah, please.
Hi, two if I may. The first one is, if you look at the clinical trial statistics for Novo over the last decade or 20 years, you've been pretty successful as an organization compared to most of the rest of pharma industry. So are you, with all this data science investment, is the focus primarily on efficiency in terms of cost, or is it on,
Yeah, I can be very specific about that. The real reason we do this is value generation. That's more important than, I mean, I'm not saying we shouldn't look at cost, but value generation, better insights, what we call asset enhancement. So the more indications, subpopulations where you see better benefit risk. Then you have time to market. Time to market means the patients have to wait shorter time. Then you have scalability. And then you have quality by design, which is not a small thing. Cost and productivity is, of course, an asset, an area we are interested in, but it's not the main driver.
Okay. And on the computational design side, I understand that you have validated your designs, 1 out of the billion or trillion you pick.
Yeah.
You in the wet lab. How have you validated the ones you rejected?
Well, we haven't because we will not go ahead and actually made that whole space. But what we will do is to collect data sets to explore protein engineering outside of the projects also. So this was a project-specific example where we use the AI, but it's important that we collect a range of data sets outside of that space to actually educate the AI on the totality of the molecular landscape, and that we are doing. So we when we train the data, use the data, we also include we will hit some of the molecules that don't work and some of the molecules that work, and that we'll use for training. But we don't go ahead for a project and reproduce sort of the whole in silico space.
No, I get that. The reason I was asking is, with these AI models, the problem is that once it learns, it's a balance between accuracy and but the thing is, if you're only feeding back successful ones, then your chemical space is getting smaller and smaller and smaller.
Yes.
Right. So, and that diversity is quite important in discovery and design stage.
Yes. Okay, but we can take it also in the break. But when we pull out, say, 2,000 molecules, we don't know which one are successful. We know that when we test it in the wet lab, and that is all feeded back. So there will be a high proportion and fraction that are the negatives.
Final question in the back.
Final question here. What would it take to see NVIDIA be a collaboration partner? Because we saw a lot of interesting names up there, but could you just elaborate on how you see NVIDIA playing in and why they're not, or perhaps how they could be a collaboration partner going forward?
Yeah, so rather than talking about a specific company, I will say we are very open to collaboration between companies. This is already happening in the Transcelerate space. And in certain areas, for instance, in the rare disease area, no single company will have enough knowledge to really do this adequately and fast enough. I'm not going to stand here and say, we're going to, go together with a company like Lilly for a variety of obvious reasons. When you move away from placebo data, you move into a space where that is not so easy. But for non-competitive, there is a big willingness, for instance, in validation of AI models or building frameworks in the non-competitive space, the Disrupt network I mentioned earlier. That's exactly what we aim to do, including some data collaboration.
But is it because NVIDIA has...
Oh, you say NVIDIA?
NVIDIA.
Oh, I couldn't hear.
NVIDIA.
Okay, no.
NVIDIA,
for NVIDIA, we won't go into details. I'm just saying.
I'm just talking about what is their capabilities.
All right, I heard Lilly.
Versus
but,
what
right.
you have up there.
You're giving me a stroke here.
So
that's a different question.
But I mean, we are talking to NVIDIA. What you saw up there is just where the companies where we have a signed agreement, and that there's a lot of companies that we are having dialogues with. And that, of course, includes NVIDIA. We would never not talk to a company like NVIDIA.
So and just imagine, maybe to close up here, that we put the power of computing that has been used in the gaming industry, or maybe even to self-driving car, to work in life sciences. And I think that is the next frontier, really, where we're going to use this. And I hope you got some really nice examples of this. But honestly, this is just the beginning of what is yet to come. But thanks for your interest. Thanks, Lars and Thomas, for a great presentation.
you.
Thank you.